Well, thank you so much for inviting me to come and speak with you today to the conference organizers. And, you know, it's really wonderful to sort of see the Bay Area phage community come together, because there really is a lot of fantastic science going on around bacteriophages and their hosts. And I'm just delighted to be here and to be part of it. So yeah, I'm going to be telling you about transkingdom interactions between phages and their bacterial host, but then also the eukaryotic host. I like starting off this talk with JBS Haldane because I think he's sort of an amazing evolutionary theorist and an interesting person. But you know, one of the interesting things that he had to say when somebody once asked him what you can deduce about the mind of the creator from his works here on Earth, he basically said that God really likes beetles because there are a ton of beetles, there's a ton of beetle diversity, and yeah, beetles. But I'm here to tell you that what God really likes is bacteriophages because phages have beetles beat hands down, both in terms of number and diversity. Basically what's going on the planet from the perspective of bacteriophage number and diversity is also mirrored inside the human body. We have, well, actually we don't really know, but by some estimates as many as 10 to the 16 phages inside the body. And, you know, historically, these have been mostly ignored because the proposition is that they affect us only indirectly. That is to say, through effects on their bacterial hosts. There are, of course, other reasons. The biology is complicated. It's been difficult to look at historically. But really, this idea of indirect interactions has allowed phages to sort of be on the sidelines, if you will. But I'm here to tell you two things in regards to this. The first is that some phages have direct effects on human cells and tissues. And then the second one is that there are particular phages that impact human health. So in the same way that you have, you know, herpes 1 virus or maybe EBV, I'm going to argue that in the future, we may very well look at particular phages and their associations with cancers or autoimmune diseases and things like this, much in the same way that we currently look at regular eukaryotic viruses. Okay, the bacteria I'm going to be talking about today is Pseudomonas aeruginosa. It's well known to many of you. I think we talked about Pseudomonas just a couple of lectures ago. But unlike the other Pseudomonas, this one is a human pathogen. It's a gram-negative bacterium. It causes a lot of skin, lung, and bladder infections. And the World Health Organization has deemed this a priority pathogen, mostly because of its antibiotic resistance and its propensity to cause chronic infections. It's a big deal financially. So in the US alone, Pseudomonas is worth about $1 billion a year, primarily in terms of critical care costs. And it's a tremendous source of morbidity and mortality as well. I'm going to be talking mostly about a lab strain, PA01, but I'll talk about clinical isolates as well. The phage I'm going to be talking about is different from, I think, most of the phages we've heard about today, in that it's not a tail and capsid phage. It is a filamentous phage in the family inoviruses. And it is, of course, long and thin, and it is lysogenic, unlike, again, the lytic phages that we've been hearing about. And the other thing that's sort of interesting about this phage is, in addition to being lysogenic, it doesn't even lyse its hosts when it gets made. So it's pseudo-lysogenic. And essentially, it can bud out of the surface of the bacterium. When it makes an awful lot of phage, the bacteria does lyse. But for the most part, it does not. And that, of course, is very different from many of the other phages we study and has some interesting, I think, ecological and kind of economic, if you will, consequences for thinking about how the bacteria relates to its phages. Okay, and so I've got two stories to tell you. The first one is that phage contributes to biofilm formation and antibiotic tolerance at sites of infection. And then the second one is that phages actually get inside of eukaryotic cells and trigger viral pattern recognition receptors that antagonize bacterial clearance. And I'm going to argue that this is important in the context of infection, but may also be part of why we or how we tolerate gut flora or vaginal flora or oral flora. And we'll talk more about that. Okay. So I mentioned that Pseudomonas is a big deal clinically, and I'm an infectious disease doc, so I spend a lot of time thinking about this bug, both in lab and in my wound clinic. And what it really likes to do is cause these diabetic foot ulcers. And then it basically will take sputum, particularly in folks with the disease cystic fibrosis, and turn it from something that looks like sputum into something that really resembles Elmer's glue, I would say. And what's doing this is basically Pseudomonas biofilms. And I think this audience knows a good bit about biofilms. But from a clinical perspective, I think about these as these thick layers of polymers and bacteria that drive adhesion, antibiotic tolerance, and actually protect the bacteria from the immune system as well. So it shelters inside the biofilm. And if anybody in the audience doesn't know about biofilms, a good example of a biofilm is the plaque on my teeth. Of course, none of you guys have dental plaque. And this is what it looks like, I guess, up close. The metaphor that I like to use when I'm going through biofilm lectures with my students is that of an anthill or a termite mound, with the idea being that the unit of organism there isn't just the ant or the termite, it's actually the entire community, including the substrate that they live in. And in that regards, it's very similar to a biofilm with the biofilm polymers. Biofilm infections are a big deal. I mentioned that you have lung and wound infections, but pretty much if anybody, or maybe your parents or your grandparents, has been in the hospital lately and got 7 days of an antibiotic or 3 days of an antibiotic, it's because antibiotics don't get through biofilms very well. And everything from your urinary tract infection, where the bacteria line the inside of your bladder, to maybe a catheter infection, where they're lining the inside of an intensive unit catheter or a Foley catheter, is mediated by these biofilms. So the question we started out by asking is, how does Pseudomonas do this? How does it make biofilms? And this Pat Secor was actually a postdoc down the hall from me when I was doing my postdoc. And he's a good friend of mine. I was just on a call with him for about an hour this morning. And one of the first experiments that Pat did when we started working together, I was working on a molecule called hyaluronan, which is a polymer. And Pat was, I forget exactly why we did this. I think he was interested in seeing how Pseudomonas uses host polymers as an energy source, maybe. But what he noticed was that when he added hyaluronan to these cultures, you got these really interesting structures that sort of showed up immediately. And when he treated these with hyaluronidase, the structures went away. And that was kind of cool because it suggested that Pseudomonas can grab stuff in its environment and organize it in some interesting structural ways. And it turns out you don't need the Pseudomonas. You can actually just take the supernatant from the bacterial cultures, and you can add that to polymers, and it essentially goes womp on the surface. And so what you're looking at over here is a glass cover slip where Pat took a pipette tip and ran it down. And so you can see that essentially Pseudomonas, or the supernatant from Pseudomonas, is able to take these polymers and turn those into a film. Okay, and of course we figured out that the key ingredient wasn't the supernatant, it was this phage. And it does this with all kinds of polymers. So this is collagen, this is DNA. So this is fluorescently labeled phage, organizing these different polymers. This is serum. So how does this work? And one thing that we noticed, this was an image that Pat took where he dropped the phage right here. And he said in the time it took him to take the picture, dropping the phage here, it had already made these structures going all the way across. That's how fast this is. And, I'm a science fiction geek. I imagine many of the people in this room are. And one of my favorite books when I was growing up was this book called Cat's Cradle by Vonnegut. And in Cat's Cradle, if anybody's read the book, there's a molecule called ice-9. And ice-9 is a crystal. And anything that ice-9 touches turns into more ice-9. And the idea is that crystals have a favorable state of entropy. And, really in the same way that a rock wants to be at the bottom of a mountain as opposed to the top of a mountain, crystals are stable. They are, this favorable state of entropy. And so this image that I showed you a moment ago reminded me of a scene in the book where ice-9 touches the oceans and the whole planet turns into ice-9. And it's really the book is a metaphor about the atomic bomb and sort of mutual destruction. But we wondered whether the structures we were looking at were in fact crystalline. And so one property of crystals is that they're birefringent. They polarize light in this direction, but not in that direction. And so they make this lattice. And in fact, if you take a polarizing light microscope or a polarizing lens on a light microscope, alginate, which is the Pseudomonas polymer, is not birefringent. This phage, Pf4, is also not birefringent. But if you mix the two, magically, you get this birefringence. It polarizes light. And you can make these really cool phase change diagrams where you have the concentration of polymer and the concentration of phage. And in the same way that you learned in high school that at a certain temperature, water turns into ice, at a certain concentration of these phages, you go from having a liquid to having something that is now crystalline. It is birefringent. And so essentially, this phage is able to organize polymers into birefringent materials. And it's not just polymers. If you look at small colony variants of Pseudomonas, which make robust biofilms, they are in fact birefringent. I think nobody's ever looked. If you look at rough colony variants, they are not birefringent. They are just slimy. And it's not just Pseudomonas. E. coli is this way. Klebsiella is this way. Salmonella is this way, all if they have these filamentous phages. They're essentially birefringent. And that's kind of remarkable. And so what we think is actually going on is that the phage is sort of a semi-rigid rod, and it has all of these negative charges down the length of it. And if that rod can rotate in space, it repels like-charged things, like-charged polymers. And so it turns out that all of the polymers in our body, for the most part, are actually anionic. And so DNA, hyaluronan, collagen, all of these things have a negative charge. And so at high densities, the phage will essentially stack these things up instantly. And if there are what we call bridging cations, you repel these things, and now you can stack these things quite densely. And so this is what that actually looks like. This is, I'm going to show you 3 minutes of footage. This was done by Yolene, a graduate student in my lab. And this is adding the phage to hyaluronan. Okay, so that's sped up a little bit, but actually not much. Kind of remarkable. And so I'm going to argue to you that bacterial biofilms are tenacious for the same reason that diamonds are forever. That is to say, they are entropically stable. And because of that, they're able to do all kinds of remarkable things from a biomaterials point of view. But so what? Right? So they're pretty, they're birefringent, they're stable. What does that actually mean? And so the first thing it means, if you take regular PaO1 and you delete the phage, which is this delta strain right here, you go from having this very robust biofilm to having this wimpy little thing that essentially falls off when you try to do scanning electron microscopy of it. And if you look at adhesions, so in a flow chamber, what we see is if you take regular PaO1 and spike in the phage, they're much more adherent. If you take a strain that doesn't have this phage, there again, now you've got adherence. If you take a strain of Pseudomonas that can't be infected with this phage, once again, you get more adherence. This is a factor that's allowing these biofilms to form and drive adherence. You also get antibiotic tolerance. And so I mentioned that your small colony variants are these bioforming unit bacterial cultures. Rough colony variants do not. And if you take the strain that we knocked out the phage from, or I guess Scott Rice actually knocked out the phage from, that behaves a lot like a rough colony variant. If you then spike back in Pf4, now it's behaving like a small colony variant, vis-a-vis the ability to resist tobramycin. So essentially, this phage is able to drive antibiotic tolerance. And this is what that actually looks like. If you take fluorescently labeled antibiotics, what you can see here is that they're essentially getting trapped in these crystalline structures. Another group whose work I really admire, the paper's Tarafter et al. They're at Oxford. They looked at this phenomenon as well. And what they saw was essentially that Pseudomonas will build itself a shell of these filamentous phages and live inside. And so the red there is Pseudomonas. The green surrounding it is this phage. And this is what that looks like under a electron microscope. So you essentially, like an M&M has a candy shell, you have this bacteria and it's surrounded by this network of phages that essentially, as I'm going to show you, protects the bacteria from engulfment by phagocytes and from antibiotics. Okay, then we developed this assay because you need to be able to measure stuff, where we took essentially the phage and polymers and put them on one side of a filter with antibiotics, and then collected what came through and put it on a different bacterium that's sensitive to many of the same antibiotics, so E. coli. And what we found was that some antibiotics are trapped in these crystalline networks and other ones are not. So aztreonam, amikacin, meropenem, tobramycin to a little extent, but actually it traps other antibiotics way better. And then there's some like ciprofloxacin that just don't care. They're not bothered by these phages or these liquid crystals. Then the question is, does this matter clinically? And so I mentioned cystic fibrosis earlier. This is actually what CF sputum looks like. And over time, it gets diagnosed in childhood. These folks have a declining pulmonary function test as measured by their forced expiration volume in 1 minute. That's what FEV1 is. It'll come up in a minute. And what happens is they get these chronic Pseudomonas infections, and over time, they get scarring in their lungs and they can't breathe. And because they get cycle after cycle of antibiotics, you have a lot of antibiotic resistance. And so what we see in the sputum from these individuals is that there's essentially a ton of Pf4. So in 1 milliliter of sputum in patients who have this phage, we find as many as 10 to the 8 copies of this phage alone, right? So essentially the phage is a solid, the sputum is a solid block of this phage. And what we did is Pat and I essentially built a polarizing light microscope and put it on a motor. And then we got a piece of software to calculate the birefringence and made that into these essentially heat maps. And if you take sputum from these CF patients, if they're infected with Pseudomonas, their sputum is birefringent, it's crystalline. If you take sputum from another individual with CF who doesn't have this phage, their sputum is not crystalline, and you can measure that. So you can take these heat maps and assign it a number, and so we call that sign D. And you see that, you know, essentially these patients who are Pseudomonas positive, most of whom have this phage, have birefringent sputum. If they're Pseudomonas negative, it's not birefringent. And if you spike back in this phage, all of a sudden their sputum is crystalline again. And then we did this study. Liz is a pulmonology fellow in my lab. Carlos runs the CF clinic at Stanford. And we looked to see what this actually means clinically. And what we found is that at Stanford, in a cohort of about 58 patients, that something on the order of 30 or 40% had this phage. Most patients did not. And when we looked at 2 other cohorts, one from Denmark and then one from essentially all the sequences from the Pseudomonas.com genome database, because again, this is a prophage, so it's in the genomic sequences, we found that globally, or in these 3 different cohorts, that probably about 40% or so of the clinical isolates carry this filamentous phage. And what's interesting is that if you look at little kids, or if you look at environmental isolates, the phage is actually quite rare. And we've done modeling experiments to look at sort of cost-benefit analysis of making this phage. But out in the environment and in little kids, only like 1 in 10 are infected or have this phage. But if you look at adult CF patients, now it's about half. And if you look at our transplant patients who've been sick for a long time, essentially 100% have this phage. That argues to me that the phage carries a selective advantage over other types of phage negative strains of Pseudomonas. And if you take, we use something called Leeds criteria to define whether patients with CF are chronically infected with Pseudomonas or not, because as you might imagine, different bacterial strains kind of come and go. And so this is a set of criteria that allows you to standardize that. And we find that essentially the PF positive patients, 100% of them meet Leeds criteria for Pseudomonas infection, whereas the negative ones, it's a much smaller fraction. And then this is kind of cool. This is new data. Essentially, each of these dots is a person. And what we find is that if you look at the ages of these individuals and you track who is PF positive and who is PF negative in this, that if you have infection, if you're PF positive with a strain that makes this filamentous phage, your pulmonary function as measured by FEV1 goes down over time. Again, this is a cross-sectional cohort. We're trying to do this with longitudinal cohorts now. If you're PF negative, your pulmonary function actually goes down much less. And so there's a cost to having this phage. And then when we looked at the sputum cultures from these individuals and the isolates that we grew out, we saw that many of the same antibiotics that are essentially the phage shields the bacterium from are the same ones that we have resistance to in this population. So it seems like this is a pretty strong selective factor for PF positive strains to exist in this environment, because these are, again, the mainstays of Pseudomonas therapy. And so our model for this part of the talk is that if you take folks with this phage, this red stuff is antibiotics. This is their mucus barrier. And essentially, the phage makes a liquid crystal that traps antibiotics and doesn't let them through to the bacteria underneath. On the other hand, if they don't have this phage, the antibiotics can penetrate better and kill more bacteria. And so we think that this is a factor that is influencing the health of CF patients. And so right now we're trying to do studies to see if we can use the presence of this phage in sputum as a biomarker to tell us which inhaled antibiotics we should give patients. So the conclusions from the first part of this talk are that Pseudomonas partners with phage to build biofilms, that this phage contributes to the pathogenic factors of Pseudomonas biofilms, and that these phages are associated with the development over time of antibiotic resistance and chronic infections in these patients. Okay. So the second story I'm going to tell you is a little bit different. So instead of talking about this phage and its bacterial host, I'm going to tell you about this phage and its eukaryotic host. And so in particular, we found that this phage gets into eukaryotic cells and essentially changes the way that they see bacterial pathogens. And what inspired this research? There's some really cool people working on filamentous phages, and one of my favorite ones is Scott Rice and Jeremy Webb. They've done great work over the years and really set the stage for most of the stuff we've been doing. And this was one example of this. So they looked at mice and essentially looked at survival. And they found that mice that were infected with the strain that lacked this phage tended to do better than mice that were infected with conventional PA01. And so we wanted to look at this, and it turns out that the pneumonia model is terrible. I don't know if people have worked with it here, but if you infect even with PA01, as I showed you a moment ago, the mice die very quickly, and you don't have a stable biofilm, and it ends up being really tough to measure. And so we needed a model of chronic Pseudomonas infection, and we didn't have one. So Yolene, who I think it's safe to say got me tenure, basically developed one between all the projects she did. And so she developed a model where the secret sauce was that we wound the mice and then we wait 24 hours. And what that allows is for the formation of what's called a paracellular matrix, a provisional wound matrix. And that is to say fibrin and all of this exudate in the wound has time to form a film that keeps the bacteria from invading into the mouse and causing it to die. And what that allows us to do is essentially take this model, and we've taken this model out to 2.5 weeks. And so if you take a relatively low virulent strain of Pseudomonas, you've now got a real bona fide model, 2.5 weeks, almost 20 days of chronic wound infection. For most of this work, we were looking at 96 hours. And then if you combine that with luminescent Pseudomonas and a machine called IVIS that allows you to quantify luminescence on the back of a mouse, you can now track infections over time. And what Yolene did is some very careful experiments where she did dose titrations and looked to see how many bacteria does it take to cause an infection in 50% of the mice. And what she found was that if you use these two strains, again, this one has PF phage, this one does not, it takes about 50 times more bacteria to cause an infection in half of the mice. And that's a neat way to think about it, because then you can perturb the system. And what Yolene found was that, again, if we infect the mice with PA01 at a dose that half of them get infected by, if you take the Delta strain that lacks that, essentially very few mice develop a chronic infection. But if you spike back in the phage, now you've rescued that phenotype. You're getting more wound infections in these mice. So how does that work? There's no antibiotics here. And so, clearly this phage is doing something other than protecting the mice from antibiotics. So what's going on? Well, one of the other things that she found was that essentially this phage protects bacteria from phagocytosis. And you take a phagocyte here, a macrophage, and you take this filamentous phage, and you take Pseudomonas, and regular PaO1 gets engulfed at a certain frequency. If you take the Delta strain, it's engulfed a whole lot more. And if you spike back in Pseudomonas, this Pf phage, it barely gets engulfed at all. And you can do this also with E. coli. You can do it with beads. Essentially, this filamentous phage shuts down phagocytosis. And how is that working? And so Yolene also did we did a Luminex panel to see what cytokines and things might be interrupted by this. And it turns out that this phage does a really good job of downregulating TNF alpha, which if there are any immunologists in the room, TNF alpha is maybe along with IL-6 and a couple of other cytokines is sort of the mainstay of the host immune response against bacterial infection. And what we found is that this basically inhibited phagocytosis until you spiked back in TNF alpha. And so what the phage is doing is it's inhibiting TNF alpha, and a consequence of that is that you don't get phagocytosis. So what else is it inhibiting? So we essentially did, we have a paper in, I think it's actually posted online now, where we looked at gene transcription in the setting of this phage or bacteria or the phage plus bacteria. And then we also looked at a Luminex panel. And what we found when we did this is that there's essentially a whole group of cytokines that are downregulated when you add the phage. So if you just look at saline, PBS, there's not much going on. There's not a lot of cytokine upregulation. If you take this phage, there's also not much going on. The phage is not particularly inflammatory. If you take lipopolysaccharide or LPS from, I think, E. coli or Pseudomonas, you essentially, as you would expect, get a lot of inflammatory cytokines. If you then put back in this phage with the LPS, the phage trumps the LPS. So the phage by itself doesn't do much, but the phage totally changes the way that the eukaryotic immune cell sees bacterial pathogens, right? And that's really cool because you can see how the immune system might discriminate between bacterial-owned versus bacteria and phages and sort of see that as two different inflammatory environments. And so what we published is that this shifts the balance between M1 and M2 macrophages. So M1s are involved in defending against bacteria, M2s do allergy and other things like this. And essentially our phage impairs the differentiation of M2 macrophages, sorry, of M1 macrophages and promotes an M2 phenotype. And I mentioned we looked at a bunch of genes. I won't go through all of that data. But essentially, this phage triggers a viral-like response in the cells that take it up. This is, I think, now on bioRxiv. And then what we found was that a lot of this is mediated by type 1 interferons. So if you look at type 1 interferon production, you can essentially get type 1 interferon production with this phage, and then you lose the inhibition of TNF in a mouse that is unable to recognize type 1 interferons. And then this is dependent on a cell surface receptor or a vacuole receptor called TLR3 that's involved in viral sensing. And we can talk about that. And so what's actually going on here is the phage gets into eukaryotic cells. I guess it's a little bright here, but DAPI is the nucleus. TLR3 is this receptor. Pf4 is our phage. And then you can see this really nice co-localization where the phage is in these vacuoles with TLR3. This is what that looks like. This is essentially in green, bacteriophage, this bacteriophage that's inside of a dendritic cell. The purple is the actin cytoskeleton, the blue is the nucleus. So phages are getting into your cells. We think they do this all the time. And they induce in the case of this phage, a TLR3-dependent type 1 interferon signature. So let me put this together as a model for what we think is going on here. So ordinarily, lipopolysaccharide signals through another toll-like receptor called TLR4 that drives inflammation through NF-kappa B. But when you have TNF alpha production, but when you have this phage present, it gets taken up and inside of vesicles triggers TLR3 and a different pathway, IRF3, that's involved in sensing viruses, that antagonizes the production of TNF alpha, and you lose phagocytosis, you get a chronic infection. Now, why would Pseudomonas do this? Why would this phage want to get into eukaryotic cells? And so we partnered with a lab at Stanford that looks at amoebas, and we actually find that a very similar thing goes on. So this is not a eukaryotic cell now, it's a amoeba. And if you take fluorescently labeled PF, it also gets into amoebas, and it also shuts down their phagocytosis. Now, we don't have all the same tools with amoebas that we do with eukaryotic cells, so I can't tell you if this is TLR3 dependent, but what I would offer is maybe this evolved to inhibit phagocytosis and essentially amoebas' ability to eat bacteria, and then it ends up inadvertently, if that's the right word, hitting us. But does this matter clinically? And so to look at this, we went to our wound model. And one of the things that Pseudomonas does is it really drives chronic wounds. I, again, mentioned I work in a wound clinic, and it's not unusual for me to have patients who have had chronic wounds for 2, 3, or 10 years that are infected with Pseudomonas. It's really hard, particularly in these diabetic patients with poor vascular circulation, to get rid of these things. And what Pseudomonas does in particular is it inhibits something called re-epithelialization. So if I'm walking out of here today and I'm in an amazing conversation about phages and I'm not paying attention and I fall and I scuff my chin, the wound that I'm going to get on my chin heals from the bottom up, fibroblasts kind of grow up, and it heals from the edges, and the epithelial cells will crawl across and meet in the middle. And so what Pseudomonas does, and this is just known in the literature, is it doesn't bother the fibroblast part, but what it does do is it inhibits keratinocyte migration, so you never get closure of the wound. So Pseudomonas wounds tend to be polymicrobial because they never seal over, and so you get superinfection with other things. And so we wanted to look at this delayed wound healing. And so another fellow who was in our lab named Gina Suh, am I running out of time? Oh, there you go. So another fellow who's in our group, she now has her own gig at Mayo. So Gina Suh does a lot of really cool work with phage therapy, and she's amazing. But she was amazing then too. And what she did is she followed 113 patients for 2 years and looked to see what happened to their wounds and whether PF might be involved or not. And so just like in our CF cases, she found that about 40% of patients with Pseudomonas about 40% of patients were infected with Pseudomonas in her cohort. And of those, about 2/3 were infected with strains that made this phage. And what was remarkable is if you look, this is what a typical wound would look like. Probably a good thing that the resolution isn't good here. But essentially, if you were infected with a strain of Pseudomonas that didn't make this phage, these are referrals to the wound clinic. So these are people who are healing poorly. It took half a year for these patients to resolve their wound infections. If, on the other hand, you were infected with the strain that made this phage, the wounds took 2.1 years to heal from the first time when they came into our system. That's a massive difference from the perspective of, what if you had that wound, or what if it was, your mom that had that wound? And so that seems like it could potentially be important. And what we did is nowadays, if you come in with a wound like this, we take pictures of it, and there's software that will calculate the size of the wound and the diameter and the depth and all this other kind of stuff. And so we use that software to essentially look at wound healing. And what we found, the red wounds are ones that grew in size, blue are ones that shrank, and green are ones that healed. These are PF negative patients, these are PF positive patients. Each of these dots is a person who is infected with Pseudomonas. And what we found is that every single one of the wounds that were red, that grew in size rather than shrinking despite our best antibiotics and most intensive care, were wounds that were infected with this filamentous phage. So I mentioned we can take our wound model out to 2 weeks. What we did is we did exactly that. We infected mice with either the Delta strain or regular PA01. And we found that PA01 led to these chronic wounds that went on and on and on. Whereas the Delta strain, they tend to resolve their wound infections by about day 7. And you see that here. PA01 strains would cause a chronic infection in this model. And then we looked at re-epithelialization on histology at the end of the experiment. And we found that, in fact, you had much less re-epithelialization if you were infected with the strain that makes the phage than the one that doesn't. We did this in pigs, and we did this because I don't have time to show you this today, but we've developed a vaccine that targets this filamentous phage, and that vaccine is under investigation in pigs. And as part of that, in pigs, we looked at essentially the effect of filamentous phage on wound healing, and we find the same thing. This is using something called transepithelial water loss. And so it measures the amount of water that can dissipate from the skin of these animals. And it's a measure of re-epithelialization. And essentially, if you're infected with the strain that doesn't have this phage, you lose much less water. You're getting re-epithelialization and a happier pig. Okay, then we did this in vitro, and essentially you can let keratinocytes form a confluent layer on a plate, and you can run that same pipette tip I was talking about earlier, hopefully a clean one, down the middle of that, and you can see it's like the world's cheapest version of a wound healing assay. You can see how long the keratinocytes take to migrate across, and if you do this, essentially, if they have phage, they're not migrating well. And this was associated with a dearth of a chemokine that was on my list earlier. I should have pointed it out, called CXCL1. Basically, if you then spike back in CXCL1, you can rescue this phenotype. I'm not sure I've got all that data here, but we just published this in Cell Reports about a month ago or something, if people want to read it. Okay, so the conclusions from this part of the talk are that phage infects human cells and triggers antiviral responses that don't cause a ton of inflammation per se, but seem to antagonize bacterial infections and the ability for the cell to defend itself against bacteria. The second thing is that through effects on cytokine and chemokine formation, these phages impair bacterial clearance and wound re-epithelialization in particular. And then I would offer that I've shown you data from CF patients and from wound infection patients. We've got data from the wound models in animals. We've got data from pneumonia models, also in animals. And so what all of these dots suggest is that you've got this filamentous phage that's widespread amongst clinical isolates of Pseudomonas that's actually responsible for some amount of the burden of disease that we associated with Pseudomonas, right? So it's not exactly herpes 1. It's not exactly EBV, but I would offer that, this is an example potentially of a model for how, Danica talked about this earlier, how your gut microbiome or your skin microbiome or the microbiome in wounds could influence eukaryotic hosts in ways that promote disease, right? So take the same model and now imagine it in terms of cancer or in terms of autoimmunity. And the point with it is that the studies we need to do are different than what you might do to look for herpes or EBV. But I think that the link between the microbiome and human hosts could very well, in some cases, be a phage. And so then the next question is do other phages do this? Is this widespread? And one of my favorite papers from the past year or so is this one here from a French group, Lawrence Zitvogel, where they showed cross-reactivity between a gut phage and I think PD-L1 or PD-1, which is a checkpoint inhibitor used in cancer. And so they basically found that this phage was blocking the activity of the checkpoint inhibitor. And so again, that's a different model for disease than what EBV or HSV1 do. But if you're the one with the cancer, it seems like it might be relevant. There are also other reports of more phages having more generalized action that depends on their number. So June Round had this paper looking at phages in colitis in TLR9 a couple of years ago. But I think we're going to see that more and more examples of this kind of thing come forward. And so let me just tie this up and get a little bit ecological, because I think that's the bent of our group here today. And I would actually offer that maybe phages are like other parasites. And what I mean by that is when I was doing my infectious disease clerkships in medical school, I had to learn a lot of figures like this. And this is an example of something called schistosomiasis. And it has a human host and it has a snail host. And then some versions of schistosomiasis have a bird host, and they do all kinds of different things. But the point is that it's very in the natural world, parasites oftentimes have life cycles that are complicated and end up having direct and indirect effects on different parts of the host life cycle or their own life cycle. And I would offer, what if bacteriophages are the same thing, where it's not schistosomiasis in a snail and a person, it's actually Pseudomonas and a phage and a person. And you have this whole network that comes together to cause disease and potentially health in some interesting ways. Okay, so this is my lab, and some of the people that did all this work, I highlighted Yolene, she's amazing. Christian did a lot of the mouse wound work. And Gina, I mentioned, is already at Mayo. She's rocking it over there. We've got a whole team of people that are working on phages. I mentioned Liz, who's been doing incredible work. Pat is at the University of Montana. He's also got his own group now. This is the team, Sandeep Kaswani, that we work on all of our wound stuff with. Chandan Sen has been doing the vaccines in the pigs. And I thank you for your attention.
We have questions.
We have a question from Zoom if we want to start with that. If you want to. So says, any sense of the genetic variation of the Pf phage and whether it matters for the data you showed? I'm wondering about genetic variation within populations of the filamentous phages and how this matters for the clinic.
Okay, so the question, just to repeat it, is there genetic variation, as I understand it, is there genetic variation in Pf phage, and what does that mean for any of this? And I would say when we started this project, we thought Pf phages were fairly conserved. And as a lot of you guys who work with phages will tell you, that's only because we didn't look at enough of them. And so when we got into this more and more and got outside of our own clinic, it turns out there's a ton of diversity in Pf phage, and that's complicated things with the vaccine tremendously. And what it amounts to is that essentially, I mentioned that the phage is a long anionic rod, and it has each part of the coat protein, which is called CoAB, has these 5 negatively charged amino acids that face out into space. And so what we see is that the exact structure of those from an antigenic or amino acid perspective varies. The structure itself is conserved, but there is variation. And we're trying to figure out what that means from a diagnostic point of view. We've had to go back and update our primers, for example, in terms of the genes that we've looked at. And there's been some really nice work that's come out over, actually, some of which has been from the people in this room, that's looked at some of the gene sequences and all of this. It's an adapting field. And yeah, so I do think it matters. It could also matter in terms of what it's doing to its bacterial host in ways that I don't understand yet. And we're excited to find out.
This is probably the obvious question. You think the phage is making double-stranded RNA inside the vacuoles in macrophages?
Yeah, really great question. So the phage, we showed, the phage gets into eukaryotic cells and makes RNA. How cool is that? But beyond that, what we actually think is going on, and so other people have shown that as well for filamentous phages, that they get into eukaryotic cells and make RNA. But what we actually think is going on is maybe a little more prosaic than that. We have a paper in review where what we find is that this works better if the phages are dirty. And so we did these experiments where we used anion purification, and then we switched to PEG, and then we switched to 2-PEG, 2 rounds of PEG to purify these phages. And we noticed that our phenotypes went away. And we did that, and what we figured out is actually the phages are decorated with bacterial structures called outer membrane vesicles, or OMV. And we could talk about why that is, but it's really cool. And so you have all these phages that are decorated with OMV, and essentially the phage is a vehicle for bringing in the OMV and triggering this. And so that's kind of fascinating because we think that, in nature, in your body, you've got, the phages are, of course, decorated with all kinds of bacterial products. And so what I would offer is the phenomenon that we're highlighting here, number 1, works for a lot of phages. And then number 2 is actually mediated by a bacterial detritus. And so one way to think of this inhibition of the inflammation due to LPS is that you're fundamentally rewiring the way that the bacterium sees or the way the eukaryotic cell sees bacterial products, because now they're being carried in on the back of a phage, and they're being directed to endosomal vesicles as opposed to cell surface receptors.